Pasteur didn't actually win the debate. The scientist he defeated was right, just for the wrong reasons. For over a century, we've told the story of Louis Pasteur as a clean victory. A brilliant scientist designs an ingenious experiment. His rival is proven wrong. Science advances. But that's not what actually happened. Pasteur won a prize. He won the argument in public, and he won the history books. But the experiment that truly ended the spontaneous generation debate wasn't his, and the man he defeated in front of the French Academy of Sciences was producing real results. Welcome to Know Science. Today we look past the victory speech. The year is 1859. Charles Darwin has just published On the Origin of Species. The question of where life comes from is no longer philosophical. It's urgent. For 2,000 years, the dominant answer had been spontaneous generation. The idea that living things could arise from non-living matter, that maggots emerged from rotting meat, that mice appeared from grain, that microbes simply formed from broth. It sounds absurd now, but in 1859 the microscope was still relatively new. Microorganisms were invisible to the naked eye. They appeared in sealed containers. They appeared in boiled liquids. They appeared everywhere, seemingly from nothing. If spontaneous generation was real, life had no fixed origin. It was self-creating, continuous. If it wasn't real, then every living thing came from a previous living thing, and that chain had to start somewhere. This wasn't just a question about bacteria in a bottle. It was a question about existence itself. Félix Pouchet was not a crank. He was a respected naturalist, director of the Natural History Museum in Rouen, a serious scientist who had spent years studying reproduction and generation. In 1859, he published a book called Hétérogénie, a 700-page treatise arguing that spontaneous generation was real, documented, and reproducible. His experiments seemed solid. He would take hay infusions, water boiled with hay, seal them carefully, and observe microbial growth appearing inside. He had repeated his experiments dozens of times. The results were consistent. Life appeared. Louis Pasteur read Pouchet's work and was immediately skeptical. Pasteur was 36 years old and already building a reputation in chemistry and fermentation. He believed microorganisms came from the air, from dust, from particles too small to see without magnification. If Pouchet was right, then everything Pasteur believed about fermentation and contamination was built on a flawed foundation. So in 1859, Pasteur began designing an experiment he believed would settle the question once and for all, or so he thought. The design was elegant. Pasteur took glass flasks and melted their necks in a flame, drawing them out into long, sinuous S-curves, like the neck of a swan. The curved neck allowed air to flow freely in and out of the flask. There was no seal, no barrier, no vacuum. But the curved section remained filled with liquid or moisture from condensation, creating a barrier that trapped dust particles and microorganisms. The curve's shape combined with this moisture seal prevented contamination while allowing air exchange. Gravity and capillary action held them in the bend. The broth inside could breathe, but it couldn't be contaminated. And when Pasteur left these flasks sitting on his laboratory bench, the broth stayed sterile. For weeks. For months. Then he broke the necks. Within days, the broth was cloudy with microbial growth. The conclusion seemed obvious. The microorganisms had come from the air, from particles, not from the broth itself. In 1861, Pasteur published his results. In 1862, the French Academy of Sciences awarded him the Alhumbert Prize, 2,500 francs, for definitively disproving spontaneous generation. The newspapers celebrated. The public was convinced. But here's what the prize committee didn't fully reckon with. Pouchet didn't stop. He kept running his experiments, and they kept producing life. Not because he was incompetent, not because he was fabricating results, because of something neither Pasteur nor Pouchet understood yet. Hay contains bacteria that form spores, endospores, structures so resistant to heat that boiling water, 100 degrees Celsius, cannot kill them. Pasteur's broth was typically made from meat or yeast extracts. When boiled at 100 degrees Celsius, these broths were effectively sterilized because the specific bacteria present in those preparations either did not form heat-resistant spores or were killed by the heating protocol. Hay infusions, by contrast, naturally contain abundant bacillus species that form endospores capable of surviving boiling temperatures. So, when Pasteur boiled his flasks and sealed them, everything inside died. When Pouchet boiled his hay infusions and sealed them, the spores survived. And when conditions cooled and nutrients were available, those spores germinated, grew, multiplied. Pouchet looked at his sealed boiled flask and saw life appearing from nothing. He was seeing real microbial growth. He just didn't know where it was actually coming from. His experiments were not fraud. They were methodological incomplete due to a biological phenomenon that hadn't been discovered yet. The man who actually closed the debate was named John Tyndall. You've probably never heard of him. Tyndall was a British physicist whose research on atmospheric dust and light scattering led him to investigate the spontaneous generation controversy. He noticed that hay infusions continued to produce anomalous results that Pasteur's framework couldn't explain, and his expertise in particulate matter made him ideally positioned to investigate why. Something was wrong with the standard story. If Pasteur had truly settled the question, why did hay infusions keep producing life in other scientists' experiments? Tyndall began systematically testing different organic materials. He noticed that hay infusions behaved differently from meat broths. He developed a technique now called Tyndallization, intermittent heating. Instead of boiling once, he heated the broth to a lower temperature on multiple consecutive days. The first heating didn't kill the spores, but it triggered them to germinate, to come out of dormancy. The second heating killed the now vulnerable bacteria before they could reproduce. In 1876 and 1877, Tyndall published his findings. He had identified heat-resistant bacterial spores as the source of Pouchet's anomalous results. Around the same time, the German microbiologist Ferdinand Kohn published his detailed characterization of heat-resistant spores in Bacillus subtilis, the common hay bacterium. Together, Tyndall's practical sterilization method and Kohn's microscopic identification of spore structures provided the complete mechanistic explanation. Between them, Tyndall and Kohn provided the mechanistic explanation that Pasteur's experiments had never addressed. The spontaneous generation debate was finally, fully closed, 15 years after Pasteur's prize. Here's the uncomfortable truth about this story. Pouchet's interpretation of his results was incomplete, not because of experimental error in technique, but because he lacked knowledge of a biological phenomenon that hadn't yet been discovered: heat-resistant bacterial spores. Every time he sealed a boiled hay infusion and observed microbial growth, he was observing something genuine. He just interpreted it incorrectly, because the mechanism, heat-resistant spores, was unknown to everyone in 1859. Pasteur's experiments, meanwhile, were qualitative, not quantitative. By modern standards, his methodology would be considered incomplete. He recorded whether growth was present or absent rather than quantifying microbial populations, which would be expected today. However, this reflects the state of microbiology in 1861, not unusual rigor for the time. His conclusions were correct for the specific conditions he tested, but the methodology, judged by modern standards, was incomplete. None of this diminishes what Pasteur achieved. His swan-neck experiments were genuinely ingenious. The logic was sound. The design was elegant. But science rarely ends with one experiment, and heroes rarely finish the story alone. We love clean victories in science. One experiment, one genius, one moment where the truth becomes undeniable. But that's almost never how it works. The spontaneous generation debate wasn't settled in 1861. It was settled in stages, by multiple people, over decades. And the man who finally closed it is barely remembered today. Pouchet wasn't wrong to keep pushing. His anomalous results were pointing at something real, something that Pasteur's framework couldn't explain. In science, the anomaly that doesn't fit the dominant theory isn't always noise. Sometimes it's the most important signal in the room. This reminds us that scientific progress isn't a single torch passed cleanly from one hand to the next. It's a relay with dropped batons, wrong turns, and winners who sometimes cross the finish line of someone else's race. I'm here to help you know how evidence actually changes what we believe. What other scientific victories do you think deserve a second look?